At Valparaiso University, which is in Northwest Indiana, I'm studying biology and environmental science with a psychology minor. And I'm hoping to go on to grad school for forensic entomology, which is what I'm going to be talking about. So this summer, I studied blowfly oviposition and the presence of predators under my lovely mentor, Dr. Bugajski. So this research falls under the category of forensic entomology, which is the use of insects in the criminal justice system. So insects lay their eggs, and then based off the development of the eggs and the larvae, you can help determine the time of death and how long someone's been dead in the postmortem interval. So this is the blowfly life cycle. So the adult blowfly here will come and lay their eggs on the carrion, which is something that's dead. And then the egg is the most vulnerable part of the life cycle to predation. And then the egg goes on and develops into 3 larval stages where the larva is actively feeding on the carrion and growing and molting. And then it goes into post-feeding stage where it leaves the carrion, and then it'll pupate, and then it will emerge as a beautiful blowfly. They are beautiful. So I had 2 different questions I looked at. First question I looked at was if there's a difference in the blowfly species and their oviposition in the forest habitat compared to the prairie habitat. And this could be something, if there's a difference, this could be something that could impact how we estimate the postmortem interval for the different habitats. And then for the manipulation study, my specific question was, will blowflies oviposit their eggs when there is a wasp present in the cup? And then here is a pretty blowfly getting ready to oviposit in one of my cups. So I had 3 types of trials. I had forest observation and prairie observation trials. And I did 9 trials total of those on 3 different trails. And then I had forest manipulations, and I had 10 trials on 1 trail. And then this picture up here is a picture of a Sarcophagidae larva. It's a flesh fly. It is a different family than the blowfly, but it is also still forensically important. Okay. So I used 2 habitats, the prairie and the forest, and for the observation. And each observation study consisted of 5 bait cups, and the bait cups was a clear plastic cup with some vermiculite in the bottom, and then a foil cup with some chicken liver that was aged overnight in a fume hood. And that would be put out in the field in the morning, covered, and then the cover would be removed 6 hours after sunrise. And then for the manipulation studies, I used yellow stance to attract the Hymenoptera, which is the wasps and the bee and ant order. And I had 9 cups total per trial, and I had 3 that were exactly the same as the observation, and then 3 that had a pinned wasp, and 3 that had 2 crushed wasps sprinkled on top. And then this is what my bait cups typically look like for the most part in the afternoon. There'd be at least a few blowflies on them, depending on what the weather was doing. And then this is my my purple prairie observation trail. So my hypotheses were that the bait cups containing the crushed or the pinned wasps will have less oviposition than the controls. And then for the prairie habitats, that they will have more oviposition than the forest and also have a greater species diversity. And then in general, I had another hypothesis that was time of oviposition and temperature will be strongly correlated because oviposition is very dependent on weather, and a lot of insect activity is dependent on weather as well. This up here is a nice up-close microscopic picture of the wasp species that I used. And then this down here is one of my largest egg masses that I got over the summer. It was really exciting. I got to see this being laid. It was made from about like 5 or 6 different blowflies all at the same time. It was really cool. So this is the yellow trail that I worked on for one of my observation sites. It's right out here behind Prairie View and the Ed building. And then I go down like a hill, and then there's the forest area. And then I worked on the red trail, which is right at the entrance where you came in today. You can see Tad's house right here. And then on the other side of the driveway, you've got this forest habitat. And then I worked on the purple trail, and this is where we are, the dining area. So it goes from behind the visitor center to the right where the yellow trail starts. Okay. So after I got eggs, I covered the cups, and then I would put them in the fume hood. And I put them in the fume hood so they could develop into the third larval instar stage. And I wanted them specifically to this stage because this is the first stage where you can identify the maggots to species. So this was one of my fume hoods. One weekend, I think there's about 40 cups that all have eggs or larvae on them. And it was wonderful. So this graph is showing average time of oviposition in hours after sunrise for all of the different trials that I did. So for the manipulation trials, which are over here, the control had an average around 9, and then the crush was around 8.8, and the pig was about 8.9. So they're all very close in the average time of oviposition. And then you jump down to the observations, and they're around 8 for both. So the average times between the two trials are different, but within each trial, they're very similar. And this graph shows the ambient temperature at the time of oviposition, and as you can see, it is not correlated at all. 0.016. So this was very surprising because insect activity is strongly influenced by the weather and the temperature outside. And then this is humidity at the time of oviposition, which is also not correlated, which is also surprising for the same reasons as temperature. This is my species diversity composition graph for the manipulation trials. I got overwhelmingly a lot of Lucilia cereularius, which is this blue color, and you'll see this repeatedly. And the only other blowfly species I got was on the crush manipulation, which is this red color, which is Formia regina. Oh, and then I got Lucilia sericata on the control. And then this green is the Sarcophagidae, which is the flesh fly family. So not that much species diversity, but lots of oviposition. And then this is for the different observation trials, like for each trail. So again, the same colors, lots of Lucilia cereularius. That's the only thing we got on the yellow forest and prairie trails. And then the only other thing we got on the purple trails were the Sarcophagidae. And then you go over to the red trail and you have Lucilia sericata on the forest trail, and then Sarcophagidae and Formia regina on the red prairie trail. So a little bit more diverse on the red trail as compared to some of the others. So when I was outside doing my data, there seemed to be a sudden shift in the time of oviposition between June and July. Like in June, it seemed that the oviposition was about like 8 to 12 hours after sunrise, and then in July, it all of a sudden shifted to be between like 6 and 9 hours after sunrise. So I don't know if it had something to do with it. It's warmer in July than June, but that was one thing I noticed, but that we did not find significantly different. Another thing that I noticed on the manipulation trials was that there were often live wasps in the cups. Whenever there were live wasps in the cups, there tended not to be any blowflies present. They would maybe be on the stand, but not in the cup, until the wasp went away. And then when the wasp went away, they would come back and kind of check it out and then do their thing. But then if the wasp came back, they dispersed. So that could definitely be impacting oviposition especially since the live wasps were mostly only on the pinned or the crushed cups. I don't think I ever saw any on the control cups. Another thing that we were wondering if it impacted oviposition was the use of these yellow stands that I used for the manipulation that I did not use for the observation. Because for the observation, I put the cups directly on the ground because it mimics the carrion, because they're normally on the ground. So we go back to this graph. You can see this average time of oviposition in general for the manipulation trial is higher than it is for the forest observation. So we were wondering if the stands might have caused that difference. And then Lucilia ceruliverdis was the dominant species found, and that's not surprising because they're usually the first blowfly species to arrive on the carrion. And then something that we could look into further at PCCI could be a future project would be to, after the first oviposition, leave the bait cups out to see if other species come and oviposit. And then this is a cute little maggot butt. This is the posterior end. And these look like eyes, but these are actually spiracles, spiraces. I forget what the actual word is. It's something like that. Don't yell at me, Dr. Buck. But this is what they use to breathe. So they breathe out of their butts, and it's really, really cool and adorable. So I would like to thank the PCCI URGE program for this opportunity, and Sarah and the rest of the staff for helping me with my project and being there with all my weird, crazy requests and questions. And then the Pierce family for the funding to do this. And then all of the other research students who helped me, like Alec and me, helped carry out all those stands, because I had 9 of them, and I'm stubborn, I like to carry them all out at once. And then my friend Hannah Thompson came up for a week and helped me, so. And then the VU Bio Department and Dr. Bugajski for their support, their help this summer. And then this is a really cool picture. I caught this dragonfly, and I had normally you have to spread the wings, but I did not have to spread the wings, and it was really, really cool.